Hey guys, welcome back to Theory Boosters, and today I will be covering Unit 2, Movement into and out of the cell. Before moving on to the lesson, I will be showing you guys the syllabus content so that you guys would have a quick idea about what will be covered. If you think that this is necessary, you may pause the video and take a quick screenshot and self-check at the end of the video to see if all the parts had been covered. Continuing on with the lesson, as mentioned in the syllabus guide, here I will be talking about three main concepts: diffusion, osmosis, and active transportation. Firstly, when it comes to diffusion, where the ideal definition would be the net movement of molecules and ions from a region of higher concentration to a region of lower concentration down a concentration gradient as a result of random motion. This definition is a key factor, so please have it in mind just in case if a question is asked, because you would need to mention all the keywords such as higher concentration, lower concentration, concentration gradient, and random movement of particles. Now, as you all know, particles are always on the move. When considering solids, particles vibrate about a fixed position. In liquids, particles slide over each other, and in gases, particles are far apart. So with this random movement of particles in all states of matter causes particles to evenly spread out in given conditions, thus leading towards the concept of diffusion. As you can see, I have listed a few examples of diffusion below so that you guys would get an idea about the concept. For instance, all gaseous exchange in both plants and animals occur through diffusion. In plants, carbon dioxide diffuses into the leaf and oxygen diffuses out of the leaf through the stomata due to the different concentrations of the gases inside and outside the leaf. Similarly, in humans, the gas exchange process in the alveoli occurs with the help of diffusion, where oxygen diffuses into the red blood cells and carbon dioxide diffuses out of the red blood cells through the capillary walls due to the difference in concentration. Not just gas exchange, but absorption of nutrients in the intestine, more specifically in the ileum, also happens by diffusion. The broken down components like glucose and amino acids diffuse into the blood vessels in the villi, and fatty acids and glycerol would diffuse into the lacteals. Even the fragrance of flowers spread throughout the area with the help of diffusion. In the diagram below, I have given a simple example with dilute and concentrated sugar solutions, which after some time would mix and form a uniform equilibrium. Make sure to remember that concentrated means that there's a larger amount of substance dissolved in a lesser amount of liquid, and diluted means lesser amount of substance in a larger amount of liquid. In this case, concentrated has more sugar than water, and diluted has less sugar particles compared to water. Moving forward to the factors that affect the rate of diffusion, which include temperature, where with an increase in temperature, the rate of diffusion always increases as particles gain energy. 2, the distance of diffusion, where there is a larger distance, diffusion would always be slower. 3, the difference between the concentration gradients, where there is a larger difference between concentration gradients, faster will be the rate of diffusion. And 4, the surface area, where there's a larger surface area, diffusion would always be faster. In general, have these 4 factors in mind, as you might get to explain certain scenarios related to diffusion where these points would carry marks. Coming down to the second major topic, osmosis. First, let's go with the direct textbook given definition of the term, where osmosis is the passage of water molecules from a region of higher water potential to a region of lower water potential across the concentration gradient through a partially permeable membrane. The keywords that you must remember are passage of water molecules, higher water potential to lower water potential, across the concentration gradient, and through a partially permeable membrane. When you guys hear this definition, you might think that both diffusion and osmosis are similar, which is true, but the main difference between diffusion and osmosis would be that osmosis only deals with the passage of water molecules across a semi-permeable membrane to achieve an equilibrium, whereas diffusion deals with any and all particles mixing with each other due to random motion in order to create an equilibrium. When it comes to semi-permeable or partially permeable membranes, which are those that only allow certain molecules to pass through them, can be categorized into natural semi-permeable membranes, which include animal cell membrane, plant cell membrane, egg membrane, etc., and into artificial semipermeable membranes, which include cellophane, whisking, or dialysis tubing. For those who do not know, cellophane is a thin, transparent sheet of regenerated cellulose, which is highly permeable to water vapor, so it's coated with a lacquer to prevent it. And whisking tubing or dialysis tubing is also a impermeable membrane that allows passage of tiny molecules, or for instance, water molecules. As shown on the diagram on the right, initially the beaker has two differently concentrated solutions, but with time, we observe the concentration on both sides have become the same. In other words, a uniform equilibrium was achieved. So in the question paper, if you are required to explain something related to osmosis and a few lines are given, make sure to write the point in full description as shown on the note. There's more water potential in the low concentration side, so water molecules move from the low concentration side, higher water potential, to the region of low water potential through the semipermeable membrane till both sides become uniformly concentrated. Another similar experiment is described below, where there is a sugar solution in the biscuit tubing connected with a long glass tube and water surrounding it. In this case, what we would observe is that the biscuit tubing would swell and become firm, and then the liquid meniscus in the glass tube would start to rise. The logic behind this observation is the fact that there is a higher water potential outside the tube and a lower water potential inside the tube due to the presence of sugar molecules. So the water molecules would travel across the whisking tubing into the sugar solution, diluting it in order to form a uniform equilibrium throughout the setup. Here again, we would be explaining osmosis under two different conditions, which are osmosis in animal cells and osmosis in plant cells. When considering animal cells and osmosis, first we will be talking about animal cells in pure water. So as you would know, when you place an animal cell in pure water, there would be a higher water potential outside the cell and a lower water potential inside the cell. Thus, water molecules would travel through the semipermeable cell membrane into the cell, causing the cell to become firm and turgid. Unlike in a plant cell, animal cells do not consist of a cell wall, thus it can only withstand a smaller amount of pressure. With the continuous entry of water, the cell would burst. The second instance is where you place an animal cell in a concentrated solution, where there would be a higher water potential inside the cell and a low water potential outside the cell. Thus, water molecules would move out of the cell and into the solution. The single term used when water leaves the cell is called exosmosis, and continuously water leaving the cell causes it to eventually shrink and shrivel. The cell membrane will get unevenly creased, and such a cell would be called as a crenated cell, as it has undergone the condition of crenation. What we must remember is that in vertebrates, the kidney maintains the osmotic balance, thus preventing our cells from bursting in or shrinking due to lack of water. Moving on to osmosis and plant cells, where again you'll be considering the plant cell in pure water first. Similar to the red blood cells in pure water, here as well, there's a low water potential inside the cell and a higher water potential outside the cell. So water molecules would keep coming into the cell, making the cell firm and turgid. Here in the plant cells, there's a cell wall which prevents the cell from bursting and can withstand the pressure that is caused by the continuous entry of water. Secondly, when a plant cell is placed in a concentrated solution, there is a higher water potential inside the cell and a low water potential outside the cell. So water would continuously leave the cell, similar to the case that happened when an animal cell is placed in a concentrated solution. When water leaves the cell continuously, the vacuole and cytoplasm would shrink and decrease in volume, causing the cell to lose its turgid pressure. Such a cell that has lost its internal pressure is known to be as flaccid. When the cell loses more water, the cell membrane would pull away from the cell wall. This condition, where the cytoplasm with the cell membrane pulls away from the cell wall due to excessive water loss, is called as plasmolysis, and this process usually kills the cell as the cell membrane gets damaged. When it comes to cells and osmosis, we come across three common terms, which is hypertonic, isotonic, and hypotonic, used in describing these solutions. Hypertonic is a term used when a cell is placed in a concentrated solution and water continuously leaves the cell. Isotonic is the term used when there is an equilibrium, where the water movement into the cell is the same as the water movement out of the cell, which normally happens in a solution which has an equal water potential as to that of the cell. And finally, the term hypotonic is used when a cell is generally placed in a solution of higher water potential, which causes the entry of water into the cell. Another subpart that is talked about under osmosis is how water is taken up by a plant. So there are two main methods where a plant takes up water, which are the vacuolar pathway and the cell wall pathway. When considering the vacuolar pathway, there is a lower water potential in the roots than that of the soil. So water enters the root cells through the osmosis. And when considering the cell wall pathway, which occurs through diffusion as the cell wall is generally fully permeable, so water passes the cell walls from cell to cell without actually entering the cell components. So as you know, we have been talking a lot about water and water potential in different solutions. So the final part under osmosis is you'll be talking about how water is used as a solvent in the human body mainly. The main role of water-based solutions such as blood or cytoplasm, which is 70% water, is the transportation of substances like oxygen and nutrients across each cell down a concentration gradient. Whatever it takes place, diffusion or osmosis in the human body both uses the aqueous media of blood or cytoplasm to transport the necessary requirements. When it comes to digestion again, saliva consists of water, which is used in the initial breakdown of large food molecules into smaller ones, both mechanically and chemically with the help of teeth. Then throughout the alimentary canal, water helps the smooth passage of food bolus and chyme. Not just that, water dilutes stomach acidity and intestinal alkalinity, thus protecting the internal cell linings. Even in excretion, water plays a crucial role for the smooth passage of feces out of the body, because lack of water at this stage leads to dry and hardened feces, which is the condition called as constipation. And when it comes to cells in general as well, water plays an important role in filling up the cell and maintaining its shape and structure, because it is always the water inside the cell that creates a pressure that opposes the external forces applied on the cell, thus keeping the cell protected. Finally, coming on to the last main topic of this lesson, which is active transport. We will go with the textbook definition of this term first, which is the movement of particles from a region of lower concentration to a region of higher concentration against a concentration gradient using energy from respiration. So the key terms in this definition are region of lower concentration to a region of higher concentration against a concentration gradient and using energy. So what we must notice here is that in active transport is completely different concept from diffusion and osmosis, because active transport occurs against a concentration gradient using energy from respiration, whereas both diffusion and osmosis occurs across the concentration gradient naturally. Some instances where active transport takes place includes during the absorption of nutrients or ions by root hair cells, absorption of glucose into the cells of the villi, and in kidneys, glucose is actively transported out of the tubules and back into the blood. So this is how the process of active transportation takes place. Firstly, the cell membrane contains carrier proteins, as you can see on the diagram below. Then ions or molecules enter these carrier proteins, and the energy gained from respiration enables these carrier proteins to change its shape and to carry the ion or molecule across the membrane and into the cell. After releasing this ion or molecule, the carrier protein reverts back to its original shape, and what you must note is that only a specific type of molecule attaches to a specific type of carrier protein. Both root hair cells in plants and epithelial cells of villi are adapted for active transportation by having many carrier proteins in their cell membranes and a higher rate of respiration to provide energy for this process. When it comes to plants and root hair cells, there is a higher concentration of nutrients in the tree than in the soil. but the plant requires more and more nutrients for growth. Therefore, the plant adopts this method of active transportation to pull in the small quantities of nutrients available in the soil. If there was no active transportation and diffusion or osmosis was to take place, nutrients would be definitely pulled out of the plant and back into the soil because of the direction of the concentration gradient. Similar scenario happens in villi in human intestine, where more nutrients are present in red blood cells than that of the lumen of the intestine. Therefore, active transportation process is used to absorb the smaller quantities of nutrients in the intestine back into the blood against the concentration gradients. So guys, we have come to the end of Unit 2. If you all think that this note which was used in the video is useful and would like to have a PDF copy of it, please visit the links mentioned in the description below. Thank you all for watching, and if you guys found out this video to be helpful, please do not forget to like, comment and subscribe to my YouTube channel. It would be a great help. Thank you and have a pleasant day.